environmental seminars for this semester, Monsoon 2020. And given the COVID situation, they're mostly going to be webinars. So I'd like to introduce Bharath and then let him get started. So Bharath will talk, and maybe Bharath can tell the viewers if he would like questions in between, or if he'd like everyone to wait till the end before asking questions.
Yeah, I prefer to have it at the end, just so that I sort of go through the whole thing and not get paused at one moment. Yeah, thank you.
All right. And maybe people can post their questions on the chat channel, so we can kind of compile them and ask them at the end too. All right.
Yeah,
so so Dr. Bharath Prithiviraj did his PhD at the MSSRF, mentored by Professor M.S. Swaminathan. He spent a total of 15 years working on microbial ecology and metagenomics. So specifically, he focuses on environmental biomonitoring, but also with a large interface with public health. So Bharath is leader for the India program on the MetaSub project. And MetaSub project, for those who don't know, studies the metagenomics and epigenetics of subways and urban spaces, and I'm sure Bharath is going to tell us a lot about this. Bharath has also worked with developing environmental sampling methods from various kinds of samples: soils, wastewater, air, and bacteria, feces, all of these. And this is in association with the work he's done with MetaSub. But he also works with several organizations to build perspectives on urban ecosystems, linking soils and microbiomes of soils with human microbiomes within a One Health framework. Bharath's really passionate about designing grassroots solutions using bioprospecting approaches. Apart from his experience in academia, Bharath has worked in the industry for many years, both in the US and India, where he consults for microbiome startups. And his experience includes working in drug discovery efforts for swine flu in 2009. And in the US, his experience spans domains such as IBS, inflammatory bowel disease, colorectal cancer, and gut microprobiotic discovery. So with this, I will let Bharath get started. And Bharath, thank you for accepting, and we're quite happy to have you kick off our seminar series.
Thank you. Thank you, Nandini. Thank you very much to the organizers, and very specifically Indian Institute of Science Education and Research, Tirupati. And again, special thanks to Professor Vijay Rao, whom I've had interactions with when I was in New York. And incidentally, he did invite me to come over in person for a talk. And due to COVID, as you know, these in-person visits are right now not a reality, but I'm looking forward to hopefully in the future making a trip down there and really engaging in person. And without further ado, let me go straight into the topic. And the topic that I selected today was keeping in mind the audience. And as such, I've been very excited by the structure of Indian Institute of Science Education Research. And having done pretty much a large-scale set of my education in India, I felt the unmet need for nurturing students at a very young stage, especially in the areas of natural sciences, physical sciences. And I think this is a very concerted effort, and the organizational structure is phenomenal, and I really attribute the think tank that went behind developing these institutes, right? So to that, I think thinking about these students and to impress upon them the fundamental areas that need to be put in focus to try to understand the magnitude of research solutions, right? So you need to go back to basics. So that's why the premise here is about our microbial cells, where I'm going to be focusing on the microbiota in the human body. And again, asking that question, which is actually a grand challenge question that people are trying to answer right now. Are humans ecologically distinct in the context of disease? And more now than ever, we are understanding the realm that we are very much part of this ecosystem that we live in. And as you know, with what's happening right now, we are basically feeding these feeder effects from the environment, directly impacting us and impacting economic development and several facets of our livelihoods, right? So one second, are you able to see my screen all right?
Yeah.
Okay.
We're able to.
1 second. Yeah, I just want to start off with acknowledgements actually, because sometimes you just miss I'm just talking about people who matter the most and people who have been influential in your career. To start off, I really want to acknowledge Professor Rob Knight, who's been my mentor since my postdoctoral research days. And the reason I'm in the field of the microbiome is because of him. And he gave me the opportunity to work in his laboratory as an early member, starting off in the Earth Microbiome Consortium project. And that was actually my first postdoctoral research project that later on became the backbone for several other initiatives like the American Gut Project and the Microbiota Global Consortium that's now becoming a global effort. Also, special mention to Professor Mark Hernandez, who I did a postdoc as well in collaboration at the same campus, and Professor Norman Pace, who's been sort of a mentor for me, and also someone I've looked up to early on when I got into the area of environmental metagenomics or microbiology. And again, Christopher Mason from Weill Cornell Medical School, Cornell University, New York, who's been spearheading the MetaStep Global Project and basically rallying around a global group of researchers to actually tackle this challenging problem. Dr. Peter Groffman, Zhangxi Cheng, Theodore Muth from the Graduate Center, City University of New York, who I worked with as part of the city effort to address the green infrastructure projects. And very specifically, I want to acknowledge Professor MS Swaminathan, who was my co-mentor during my PhD, and he's the founder of MS SRF, as you know, Dr. G.N. Hariharan, who was my PhD advisor, and the director of biotechnology, MS Swaminathan Foundation, Chennai, and several other collaborators, Rich Shaw, specifically USDA, New Jersey, Dr. Kelly Craven, Noble Research Institute in Oklahoma, James Sears with Terradome, Colorado, global collaborators. At this time, none of this research would have been possible, especially the components of what I'm going to speak about, without these global consortia efforts from global collaborativeness. And the consortia, as well as the funding organizations such as Gates Foundation, Sloan Foundation, and the nonprofits that I am engaged with in terms of scientific advising capacity, like the Small World Initiative and Urban Soils Initiative. So let's jump on. I just want to impress upon this is basically for students. Yeah.
Sorry, before you go on, your voice is fluctuating a bit. Is it possible for you to maybe have that device a little closer to you or something?
Sure. Let me just go on to the basics here. I'm just going to do a plugged-in earphone. Maybe that will work better. Let's see. Is it okay now?
Yeah.
Okay. All right. Let me just take this off. All right. So so if this works, that's good. This is for the first or second year students to impress upon you in terms of the tree of life. In terms of, it's one of the most fascinating images that especially as a biologist, that you can go through, which is trying to cartographically represent the tree of life and the domains of life as you would. As we know that we evolved since millions of years ago when Earth was born, and after several of these events that involve mass extinctions and several ice ages and where not, right? So you basically evolved from bacteria, these organisms that have survived the test of time, the vagaries of all these stresses and perturbations. And at this end of this radiation, or at least not the end, the perceivable omnipresent that we are in right now. In this particular figure, I found this figure very compelling, the way the illustration has been put out. And actually, this figure only covers the organisms that are in existence right now, and it doesn't cover those that went extinct. So it's interesting to see the amount of radiation that you find from bacteria, archaea, and moving on to eukaryotes. And I just want to bring up the fact that, especially with mammals, you're looking at these groups starting off from platypus all the way coming down to human beings and the old world, new world monkeys, the apes, notwithstanding that, right? So you also have interesting mammals in this list that are, again, one of the most debated right now, including bats, armadillos, or basically anteaters. So these are also ones that are in contention right now that people are talking about. And when we think about evolution, we also need to think about the time that we are in right now. There's this staggering figure that came out in Science 5 years ago, this paper by Stefan Ambo, Human Perturbation of Planet Earth. And some of these areas that stood out in terms of high risk were genetic diversity and nutrient flows, as well as the biosphere integrity. So these are some of the elements that
are actually contributing to the events that we are seeing unravel in front of us right now. And it's the lack of genetic diversity because of environmental degradation, environmental mismanagement that's led to several of these crossovers in terms of species occupying territories that otherwise they would not inhabit, or encroaching into their territories so that they basically don't have a habitat to get to and have to have interaction with human hosts. So that's actually leading to these spillover events. And when we see this coming, it's sort of the overarching question that we would want to ask. And Lancet had this review paper that came out close to 10 years ago from this fascinating group called EcoHealth Alliance in New York City. It's actually a nonprofit that works on pandemic research, and they've done phenomenal work over the past 2 decades on trying to model several scenarios associated with pandemic or strain dispersal or exposure from these zoonotic, in terms of the zoonotic diseases from these vectors, in terms of transmission, and also from these animal hosts. So this is basically on the y-axis, they have mapped out the number of cases over several years and spillover events coming out of wild animals, domestic animals, or in this case, the human amplification scenario. So it sort of gives you a moot point to understand that we are forever at risk of exposure from these spillover events because of animal incursion or us basically encroaching into animal territory. And this is actually a staggering figure that recently came out in Nature reviews. And it talks about COVID-19's impact on systems across the globe, right? So if you think about it, the impact is magnified by several folds in terms of the areas that it can go and affect, starting with agriculture. Obviously the first point that we need to consider is human health, which is the most direct impact that's going to cause morbidity or mortality. But at the same time, you have these peripheral effects that are going to affect different other components of the ecosystem, starting with agriculture or the way you produce food, that's going to affect productivity, and in terms of economic activity, which is something that's also trickle-down effect. You're going to see jobs affected, demand for energy increase because people are going to stay indoors more longer. That's also going to cause challenges to an individual staying indoors. Again, this is also created by the dynamics associated with how climate vagaries can occur. And those vagaries can also affect the ecosystem around us, which is going to affect animal species and cause these catastrophic events in the environment. So it's something that we need to take into consideration. So as we think about these drastic effects that COVID is causing, in a very subtle manner, there's something that's going on in the background. And this paper in New England Journal actually captured that in a sort of a very succinct way, where Bach et al. Talked about the incidence of infectious diseases over the past 5 decades, backward from 2000s. And if you look at that, the infection diseases such as rheumatic fever or measles, the mumps, or TB, in terms of the rate of decrease has actually been drastic, but I wouldn't put that number consistent for South Asia, specifically India, because TB is still a challenge that we are facing, and it's a challenge actually in parallel with COVID that's actually going to become an obvious challenge. So at the same time, they're also seeing a spike in the incidence of immune disorders, and these disorders include conditions like asthma, type 1 diabetes, multiple sclerosis, and Crohn's disease. And if you look at these conditions, each of these conditions have an attributable facet connected to microbial dysbiosis or imbalance in the microbial entities that colonize humans. Basically, us as an ecosystem, we have microbes that colonize inside of us and on us, and are also able to mediate our functional capabilities in this case. So this is exactly what we constitute or call the human microbiome, which is essentially the genetic makeup of microbiota that colonize us. And in terms of the numbers, right, it's a staggering number of these microbial cells that you find in our body, which is approximately close to 100 trillion cells. When you compare that to human cells, it's like 30 trillion cells. And at the same time, in terms of microbial genes, we're talking about close to 2 million genes. And in terms of the human genes, it's actually minuscule when you talk about these numbers. So this image comes out of that paper in 2012 from the Human-microbiome project that NIH published. That was actually the tipping point for several other initiatives to follow suit on trying to understand the magnitude as well as the directionality of what the numbers of these microbes mean, and at the same time, in terms of functional attributes, what they convey to us or what we need to do about the signal that they convey to us. So as the human microbiome brought out this data, it basically provided us with a blueprint for what we can call the exposome within us. In this case, I define the exposome as the microbial cloud, or if you want to call it in a very spiritual way, an aura around us, which is constituted by microbes. And largely, the foremost abundant phyla that they found from the data that they derived from this effort was these groups of phyla called actinobacteria or Bacteroidetes or Firmicutes or Proteobacteria. Some of these phyla names are something that I'm going to repeat often during the presentation. So just it's going to kind of recur in this pattern. But at the same time, they found a low abundance phyla from these microbial groups that they were able to locate in very specific niches as in locations in the human body. So if you take, for example, they had differences that they could see between stool or fecal samples, the microbes that constitute the fecal sample, and ones that you find in on the cheek or on the tongue or on the nose as well as the skin, which is actually very different from each other. And just taking an example, for skin microbes, one of the most predominant ones we found was, especially nasal microbes, they found Propionibacterium acnes, which incidentally is something that lives in the skin as well as in the nose of people and causes acne in individuals who have certain immune imbalance, as well as certain other added conditions associated with that. It could be conditions associated with imbalance in body mass index and things like that. At the same time, there were also species in GI tract that were very interesting that were dominated by these species called Prevotella copri, which is very interesting and it's something that I would go into further on during my talk. And again, as a constitutive fact, microbes make up close to, if you think about a person weighing 200 pounds, close to 3 to 5 pounds of that body mass is going to be microbial. In this case, it says bacteria, but of course, we are going to talk about the differences of types of these microbes that are colonizing us. So again, how did this all start? And how do we even think about examining microbes in this way? It sort of was spearheaded by Professor Norman Pace early on in University of Colorado Boulder, initially in California, Berkeley. Then he moved to Boulder, and Norm actually started investigating extremophiles or organisms that survive in ecosystems or environments that are very highly fluctuating in terms of their basic environmental variables. This could be variables such as pH, temperature, or relative humidity that actually make them very interesting. For instance, he even studied microbes in the Yellowstone geysers. And basically, he also went into the microbial mats that were found in the Pacific, areas like Baja California. And also, he went into these hydrothermal vents, or actually, he actually collected samples from these areas and looked at these microbes from these very, very hyper-diverse ecosystems. And he did that using a technique called environmental metagenomics of biomarker genes. And his approach was to look at bacteria and archaea using the conserved gene for 16S ribosomal RNA, which is highly conserved between bacteria and archaea. And they have, as you know, like three subunits. Basically, you have subunits, you have different types of these ribosomal RNA, and very specifically, 16S was chosen because it had close to 9 hypervariable regions that you could actually query at different instances and try to understand whether certain regions were drastically different between each other. When you're pulling these sequences out of samples and you're asking the question as to how much of similarity is there between these hypervariable regions, which means these species are closer in aggregation to each other, or if there is a variability, there are going to be differences in the sequence trees that you're going to see. So this was actually the initial efforts, but at the same time, we need to be cognizant that for fungi, that's actually the other, also in the eukaryote domain, you had the 18S ribosomal RNA that could be used as a biomarker gene to examine, or the internal transcript spacer region that is also, that's something that's a gold standard that's being followed. So how do we progress from there? So because of the advances in terms of sequencing, just the way Anton von Leeuwenhoek found the initial microbial cell using a simple microscope that he devised, these days we have the ability of technology that's even more robust to help us kickstart or amp up the ability to span across several depths of the sequencing data that you can pull out. And you have different versions of these sequencers, starting off with the Illumina HiSeq, which was in the early 2010s, and they were pretty robust in the way they were able to churn out data, and also different other companies that are able to both provide enough throughput And also, they're able to miniaturize the technology in a way that right now you have Oxford Nanopore handheld sequencing options, which has actually been used at the International Space Station. If you think about it, it's pretty staggering that you can even collect a sample in space and pretty much sequence it in that particular location, which actually gives us the option right now to have a handheld power of going out into the field or wherever you want to go sample and do, running out your sequencing experiment, right? So it's something that was unheard of even 10 years ago when we guys were starting off in this area of metagenomics and NGS. So it's really compelling and really motivating to see that. Notwithstanding only sequencing for DNA-based approaches, we have concomitant approaches that could be adopted in this case. So this is with techniques such as the ones I spoke about for biomarkers. We can also expand that to what they call metagenomics, or looking at the whole genomes of these microbial entities. And there's a technique called shotgun whole genome sequencing, where you're pretty much shooting into pockets on the genome of these microbes and deriving information from genes from these pockets, and also using bioinformatic approaches to stitch back these pockets of genome entities that you have at hand, and you're thereby able to resolve information that you can interpret and so on. And also there's RNA-based approaches where you can actually look at the functional robustness of how these organisms are responding to certain perturbations of signals, and at the same time, what sort of pathways are being activated in these organisms at a given time. So that's the way where you are able to resolve or basically ask questions on specific enzyme dynamics or interactions on the functional level and how that works through. And right now, we're also in the realm of understanding the power of protein interaction and through a metaproteomics approach, which is actually also an omics-based solution at hand that's actually progressing pretty fast. And of late, there's also this element of understanding how the functional interaction occurs in terms of co-evolution paradigms or co-interaction paradigms between microbes, which is where metabolomics comes into play. So we pretty much have these three or tripartite or four directional ways of interacting or understanding data that you generate from microbial environments, which is actually making everything more robust in the current time. So because of the capabilities with sequencing that I pointed out, now we are at the stage where we can discuss the new tree of life. And Jill Banfield's group from Berkeley came out with this paper a couple of years ago, led by Laura Hugg, and they put out data from whole genome-based approaches. They actually did the same stitch back approach where they created these shotgun genomes and they were able to stitch back on genome data. At the same time, what they came up with was actually a very staggering set of information, that they found that archaea are a group that intersperse eukaryotes within them. So they found that you have 5 of the eukaryote supergroups that were branching within a superphylum. And they define 92 bacterial phyla, 26 archaeophyla, and eukaryotes interspersed in them. But at the same time, they still saw a lot of uncultivable candidate phyla or taxa that were dominating. In this case, this is the lower panel of this figure, which actually gives us a clue as to how much of this is not yet discovered in terms of not yet resolved. It's still out there, and there's further efforts that need to be put in place to resolve that. So the human microbiome comprises, as you know, bacteria, fungi, viruses, as well as parasites. And you can define them as microbiota when you talk about parasites. These extend from the GI tract to oral cavity, or the other way around, the vaginal mucosa to the skin, respiratory tract to our oral cavity, or circulatory system of late to the urinogenital tract. And more specifically right now, the viral microbiota is something that we want to be talking about because COVID is going on. And if you think about the first efforts that went into the space of virome, I'd like to acknowledge the work done by Forrest Rower at San Diego State University, because his group was actually the first group to put together virome-seq assembly or cross-assembly phage data, 2002, from marine organisms, marine bacteriophages. And that actually resulted in data being specifically generated towards a virome database, which is actually one of the unmet needs that actually huge lacunae in the field of the microbiome. There's not enough data generated from viruses. There's more data from bacteria or archaea. There is something intermediary when it comes to fungi, but with viruses, it's really, really low. but hopefully this is going to change now with what is happening. And if you think about viruses alone, there are close to 380 trillion viruses. And you compare this with bacteria, it sort of brings us to the more point of asking who is actually keeping us healthy, because we know that these organisms coexist in our bodies, but we know so little about them. And it's actually staggering that we don't know enough about them in a way that we can't even resolve them. And just the statistic number that actually the USAID Predict project put out, if you think about 700,000 viruses, 24 viral families, they all have the potential to cause human infection. And again, as of now, they've identified just 111 of these viral families, and you basically are having close to 650,000 to 840,000 viral species that are having the potential to be a human pandemic or epidemic proportions, right? So that's actually one of the huge, biggest challenges that we're going to face. And it actually holds good for viruses like HIV or other retroviruses. So this is something that we need to be thinking about. And at the same time, as we think about these threats, we need to address what our strengths are in terms of what we innately have, along with, of course, our immune system. That's our first line of defense. We are encompassed by these set of bacteria or microbes in our bodies. And how are they defined inside of us, right? As you would know from the research that's been done, these microbes are very seasonal, or they respond to seasonal shifts in our preferences for diets. And from time immemorial, we've seen that the microbial diversity over several thousands of years ago has depreciated in their numbers as well as in the diversity that you would find in current day. This is because of the domestication process or the advent of agriculture as a practicing facet to how we eat our foods, from being a hunter-gatherer. And several of these have become reductionist approaches where at every point in time we are reducing the diversity of microbes that can translocate into our systems. And again, right now, I want to reflect on the study that came out in Nature in 2011 by Arumugam et al. And they defined these microbial enterotypes, or these flagship groups that you would find, keystone groups that you would find in the human gut. And they determined that long-term diet is influencing these groups of microbes. In this case, these are phyla such as Bacteroides or Prevotella or Ruminococcus. And each of these have a specific trait associated with them. Prevotella species, as it says, is very specific to people who consume a lot of vegetarian diet or complex carbohydrates, which are closer to the hunter-gatherer diets. At the same time, folks who consume Western diets that include people who eat meat, who have industrialized Western diets, that's something that basically is going to predominate in their gut. At the same time, you have fiber-rich diets that are constituted of species like Ruminococcus. So if you think about it right now, we are basically at a scenario where we can think of the East versus West in this case, right? The East, people in the East are predominated by this group called Prevotella, and people in the westernized countries are predominated by groups in the phyla called Bacteroides. And it's pretty much driven by diet, which is actually staggering if you think about it. Again, there's this biggest gradient that you find in the human gut, which is actually represented in this figure where they're actually spatially or habitats in a very habitat-specific manner demarcating these boundaries. And if you think about the stomach, it's highly acidic and it's oxygenated, and you have a very limited load of bacteria that you would find in these habitats. Again, they are predominated by very specific species like Firmicutes or Actinobacteria. And the duodenum, again, you get to a pH which is neutral. It also has an interference effect from bile acids or mucus or the sugars or mono and disaccharides. At the same time, there's a lower level of oxygen availability, which actually results in an increase in bacterial load. And also you have a variation in the predominating type of microbes. And it's sort of lower in diversity, again, because of the flux caused by the pH. And when you go into the gut, into the colon or the large intestine, you're going to find a higher bacterial load predominated by microbes that are actually forming groups that are non-oxygen dependent, which pretty much raises the scope for these anaerobic entities. And those are highly specialized that help you digest indigestible polysaccharides or complex sugars, which actually is very important for the gut to render these microbial services, microbiota services. These services span several areas, starting with immune response, with metabolism, contributing to nervous system or optimization for the gut-brain axis. So if you look at this figure that came out in Sandberg's paper, Science, it basically gives you a representation of what happens when there's a disturbance effect that happens in the microbiota, right? You're going to have depreciation or certain disturbances that happen, critical variables. For instance, these categories like digestion or calorie storage or inflammation are all going to be either increased or decreased based on the stress that the gut is facing. So several of these aspects are very critical for optimal functioning of the gut, as well as optimal maintenance of the human immune system. So in a way, what is happening right now with these diets or with these modern diets is we are overfeeding on these modern diets, or actually indulging in these sugary diets that we consume, but at the same time, we are starving our gut from within, which means the more and more you consume processed foods, carbohydrate-rich foods, or sugars, the more and more you're depriving your microbes that co-occur in the large intestine or the colon. In this figure, what they're showing is you have what you call microbiota accessible carbohydrates. And these carbohydrates constitute the groups that form the complex polysaccharides or the complex carbohydrates, such as mannose, glycan entities, that actually are required by the large intestine microbes to perform optimally, especially by producing these short-chain fatty acids. And short-chain fatty acids are very important for the body to maintain homeostasis. And this is something that's of late been unraveled from some of the metabolic or metabolite-based approaches that have been done in studies, in disease system-based studies. And again, when you think about a scenario on the bottom, it's basically consuming simple sugars and carbohydrates or sugars that's basically going to get processed in the small intestine. And at the same time, the microbes in the colon do not receive the ingredients that they really need to perform optimally. So we are having a continuous set of these disturbances that are occurring, right? With the diet, with sanitation that's actually overtly practiced, where you're following extreme higher levels of sanitation and not even exposing your kids to be in the vicinity of soils, and thereby not helping them train their immune system by exposures to such otherwise environmentally available pathogens, or commensals for that instance, commensal groups which can actually work in unison with your existing flora of microbes. And at the same time, overconsumption of antibiotics when it is actually absolutely not required in situations such as this, where people have actually overdosed on antibiotics thinking that that can save you from getting infected with COVID is actually illogical, right? And again, healthcare practices, which include inadvertently promoting C-sections for pregnancy, and also recommending baby formula, food formula, right? So from an early stage where you're depriving the child from deriving those natural microbes from the mothers. But the reason I brought up fermentative microbes here is because recently, through the data, they were able to unravel that you have specialized endurance traits that you can derive from these select microbes. And there was a study, very cool study done in Harvard Medical School by one of my colleagues, actually collaborators, Jonathan Scheinman, from Professor George Church's lab, where they studied a species called Vinella atypica. This is an anaerobic microbe that pretty much is able to survive through the fermentative pathways. In this case, it produces basically the microbe Vinella atypica, which is found in the gut of these elite marathon runners from Boston, were able to utilize the lactate produced during exercise. And that is actually used as a substrate for their metabolic consumption, thereby reducing fatigue that would set in from endurance activities. And this is actually staggering information, because they were able to replicate this in mice models, where they fed these mice strains of this particular bacteria, and at the same time, they also simulated conditions for endurance. Basically, they can make these mice run on treadmills that have been designed for mice, and at the same time, they can also calculate or study metabolic output from these mice on how much of these short-chain fatty acids they produce, how much of lactate they're producing, or lactate they're producing that's getting metabolized doing that particular endurance activity. So it's clear to us that the solution, or actually one of the solutions that we would have to find a way to promote optimal performance, both at the level of fitness and at the level of the level you can cope with the environmental stresses is potentially hidden in your gut. And the reason that is there is because this is being conserved from several generations. I talked about this, actually, this idea became a product, and the product became a company called FitBiomics, and I had an opportunity to work with them in a short time with this group, and they're doing some really fascinating work on developing the next generation of probiotics from athletes, elite athletes, that potentially a common person can also consume. But again, it's hypothetical to see whether everybody has the same fitness level as someone like a Usain Bolt or someone who's performing like a Michael Phelps. So it's a moot point. So it can be very, very location specific, and you need to have approaches that you can optimize for different ethnicities, right? So I talk about ethnicities, and ethnicities brings us to an interesting question. As an ethnic group, we all have certain traits that are inherently earmarked for these groups, right? So they ask some of these questions associated with the microbes, whether if, for instance, you have a group that moves on from one continent to another, what happens to the microbes that are inherently there in these individuals? So this is a study done by the University of Minnesota, a group led by Dan Knights, who was my colleague from Rob Knight's lab, and they did this cool study in Thai immigrants, groups of Thai people who have immigrated to the United States over 3 decades, and people who have been practicing the native diet coming from Thailand, and people who are in that same cohort who moved to the US and have followed an adopted process of consuming processed foods or Western diet. So what they found out was you have groups called vanished taxa, or otherwise called as volatile or associated negatively with these industrialized societies of humans, which means there are these groups of microbes that you tend to lose if you go to a westernized habitat where you're compelled or you are in a way influenced to consume these diets, thereby losing the traits that those beneficial groups are performing in terms of function for you. So you had a trade-off between, in purple as explained, Prevotella strains, which are very efficient at degrading fibers or fiber-degrading enzymes that would deprive in these populations that are growing up in the US, and at the same time they're exhibiting an increase in their BMI. Thereby they are actually presenting obesity in these individuals. The same individuals whose parents or whose ethnic lineage is from that particular region of Thailand. So they actually did several efforts on trying to understand why this paradigm is. And off late, there's also this approach where the Native American people in the US are adopting to their own Native American diets rather than accepting food packets from the federal government, because that includes these processed foods. They have an awakening moment where they want to know that they want to preserve their culture, the dietary culture, as well as that's something that's actually going to help them preserve their health in that sense. So since we talk about the immigration aspect, we also need to think of the globe and the urbanization gradient that could be explained perhaps by the microbes that are constituting us. And this paper explains in a nutshell, based on GDP, what is the distribution of microbes that you would see across these spatial habitats. So in this figure, in purple, like basically they have distribution across gastrointestinal tract, the skin, the urinogenital tract, and the oral cavity. I'm going to take 2 examples in this case, starting off with India, where in the gastrointestinal tract, we have Prevotella dominance. This is coming from the sparse data sets that are available in India on the microbiome side, and I know that the Indian gut microbiome project is sort of ongoing, so that's going to unravel interesting information about the current generation. And at the same time, if you look at a country like the United States or Canada, in this case, the dominant group in the US is Firmicutes, which is actually a phyla that's associated of groups that are implicated in obesity, right? And if you look at some of the similarities between India, and take an example from East Africa, countries like Malawi or Tanzania. I'm taking an example of Tanzania and Malawi because they are East African, and that's also, biogeographically speaking, we were continental landmass that were merged together at some point in time. So you can see the same set of groups that are predominating these countries. Very interestingly, and some of the early hunter-gatherer groups originate from that particular region of East Africa, which is actually a moot point. And again, it's interesting to see the rate of urbanization having an influence on how the disparity is with the diversity of these microbes that you could see in real time. So with that in mind, right now there are efforts from global scientists to put together a map, or in this case, a unified genome map, constitutive of these approaches that I talked about, where they do metagenome assembled sequencing or metagenome assisted genome assembly, metagenomics assisted genome assemblies, and put together several genomes from around the world. So right now, from the human gut microbiota alone, you have 204,938 reference genomes. The staggering study came out very recently from Nature Biotech, led by Alexandre Almeida, and some of our Metasub collaborators are on that paper, including Nicola Segata from Italy. And they actually have generated isolates from human gut microbiota and also these metagenome MAGs, or metagenome assisted genome assemblies. So if you look at it from the United States and from China, you have a predominant number of these genomes that will be generated because of the robustness of availability of sequencing or access to sequencing. And even some of the Scandinavian countries, like basically you have European countries like Spain, UK, and Denmark, Netherlands, as well as portions of Scandinavia. So these numbers dwindle down as you go through the other continents. And actually, if you look at India, they're in the 101 to 1,000 category. I think it's a matter of time before these numbers rise. But this is a huge resource for people or for researchers who want to progress in the stage and ask questions on what genome contributions are there within the human gut and how you can inform understanding of disease from these signals that you can generate, along with, of course, the magnitude of the humongous human genome data that's available and that's been trained, and a lot of the other research has happened since now. So because of such robustness in data, right now what has happened is you have a phase 2 of the Human Microbiome Project, which is called the Integrated Human Microbiome Project, that specifically looks at disease models. They look at disease models starting with preterm birth, or looking at maternal microbiome, maternal vaginal microbiome, and looking at type 2 diabetes as well as inflammatory bowel disease, which constitutes 2 conditions like ulcerative colitis and Crohn's disease. I'm going to focus specifically on IBD in this case, very specifically on the example of Crohn's disease and ulcerative colitis. So this is one of the figures that came out of that paper. This is led by Lloyd Price. I actually forgot the citation here. Lloyd Price and Kurt Sutnover's lab in Harvard, MIT. And from the left panel, if you look at the density of the taxonomic groups of bacteria that you find in these individuals, in this panel, in blue, you have the non-IBD cohorts, and in in pink, you have the Crohn's disease cohort. In pale brown, you have the ulcerative colitis cohort. So that is actually a group of non-dysbiotic, which means they don't show an imbalance in the microbial communities. And also the separate categorization is with dysbiosis, which means they show a disturbance, obvious disturbance in their microbial feedback. So some of these species are actually flagship species that you would find in the gut, starting with Faecalibacterium prosnitzi, which is actually an indicator bacteria, if you will. It actually is indicative of homeostasis in the gut. And in some of these individuals who otherwise have an imbalance in that are basically going to have a challenge with how you would interpret their profiles. Again, you also have fermenter microbes in this group, like Ruminococcus naevis. Oh.
I think we lost your slides.
Yeah, I think we lost your slides. Can you please check?
Is it right? Is it now? Is it good now?
No.
Sure.
1 second. Let me try it again. I think from this screen I got logged out, which is weird. Is it?
We can see you.
You may have to share the screen again.
Yeah. Is it?
It's coming up.
Okay.
Yeah, can you explain this slide? I think we lost it when.
Sure, yes. This is coming from the second phase of the Human Microbiome project, where they very specifically looked at 3 disease models, starting off with preterm birth and the maternal microbiome, and type 2 diabetes, as well as the inflammatory bowel disease, specific to ulcerative colitis and Crohn's disease, which are 2 types of these IBD specific syndromes. And in the figure onto the left panel is the representation of relative abundance distribution of very specific and significantly varying metagenomic species. In this case, this is constituted of close to 1,500 of samples that were collected from 130 of these subjects. And in terms of metabolites, again, there were close to 546 samples that were derived from 106 subjects. At the same time, this was compared to samples that were coming from dysbiotic individuals, wherein they are exhibiting a response, a disturbance response from the profile of their microbiota, to the profiles of people who did not show symptoms in terms of the readout that you see from their microbiota. And at the same time, what was interesting in this case was they were showing obvious differences in some of these communities. When you look at the blue panel, it's the non-IBD cohort compared against the cohort that's having the disease. And you can see changes between some of these indicator microbes, like the one in the first category, Alstipus, which is actually involved with processing of proteins in the large intestine, and also some of these species like subdolic granulum, which is actually something that is involved with branched chain amino acid metabolism, and some of these species like Rosiburia, which is actually an indicator of stable gut profile, along with F. protsnitzi, which is Faecalibacterium, which is also reported to be one of the homeostasis sort of representing of the balance in these microbiota. So they're able to pick up on spatial signals from these groups of cohorts, very specific to disease, but at the same time, the change in the ecological specificity for these strains is actually giving them information, further information. But again, the functional readout from these microbes is actually critical to understand to what extent that variability is explained. And for that, what they did was to use metabolomic approaches to measure short-chain fatty acid production capabilities from these cohorts. And there is a specific decrease in these butyrates and propionates, which are short-chain fatty acids, that actually act as precursors for helping, like I explained, circulation in the colon, and also helping maintain the optimal level of cytokines that thereby can regulate the rate of inflammation, which actually is very critical when it comes down to that part of the gut. And I'll explain in my next slide why that is critical. And concomitant with that, they also had serological markers that are pretty much used as titer responses that they study for conditions like Crohn's disease, where they look at these ANCA antibody profiles as well as IgG or IgA profiles. And along with that, they have the anti-outer membrane protein profile that they see for some of these subjects. And they had the profiles for what they compared with the Crohn's disease patients to the normal cohorts. And if you look at these antibodies that such as the ones in IgA or IgG, they were highly represented in their titer when compared to the baseline. And that is actually something that they showed for the first time in a very concerted manner, where the way the population was selected and the subsets were maintained was actually done in a very robust manner, and the clinical trial was established, and thereby they were able to show this particular data in a first of its kind manner. So this is one of the models for IBD, but at the same time, off late, they are able to show signals associated with inflammation and how that correlates with the dysbiosis in microbiota. And this has direct implications in disease models such as colon cancer or colorectal cancer, where there is a role for translocation or spatial differences that exist between these regions within the gut that constitute the lumen or the mucosa, which is the intermediate layer that lines the intestinal walls, or the submucosa, where you're having interaction between microbiota and pathogenic bacteria, as well as interaction in the mucosa that includes the immune cells. So we have these environmental factors that contribute to changes or disturbances that can occur in these regions. But at the same time, there are dietary factors that also have a role to play here, because the mucus region that line the intestinal cells is very critical in a way that it provides that barrier, or what you could call the, just similar to the blood-brain barrier, you have a gut barrier or a gut pathogen barrier that needs to be maintained for optimal maintenance of homeostasis. Otherwise, you're going to have inadvertent events that are coupled by the genetic factors that actually predispose you to exposure, also to the potential for a cancer cascade to actually follow through. So there have been several studies that are ongoing. There have been some really hallmark papers that have come out in this area of colorectal cancer, led by groups in EMBL, Pierre Bourque or George Zeller, just giving some references, and then from Paul O'Toole's group in Ireland. I'm not diving deep into some of those studies because I need to move forward here, but there are certain really interesting areas that you could certainly explore, look into. And also this other conundrum that we are faced with when it comes to conditions like gastric cancer, which is H. pylori. Again, knowing that it's sort of an existential group that you find only in the stomach. And again, when you have dysbiosis, there's this potential for other microbiota to seep into the areas like the duodenum or the stomach, thereby causing inadvertent co-occurrence or interactions or trade-offs between H. pylori and these microbiota that can also lead to disturbance events such as what you're seeing in conditions like gastric cancer. This is something that clinicians are also looking further into, but there's a lot of ongoing research on that aspect. Again, coming back into the seasonal fluctuation aspect. So there was a study again done in Stanford where they looked at these hunter-gatherer groups in East Africa, specifically the Hazara tribes. And from them, what they were able to find out was you can define these traits in microbes within us based on our occupation and our diets. They took this example from a study done in Nepal, where they categorized groups of people who constituted foragers, which means people who go out to collect the food, people who are in settlements, but then they accumulate food from where they settled in, or people who go out into the field and practice agriculture. And they map them back to these industrialized communities. And if you look at this, in this case, this is a relative abundance heat map that you can pan back into. You can see that very obvious shift in these Danish taxa, which is led by these groups dominated by Prevotella, which is the fiber-loving group. And again, you have groups such as Succinovibrio or Alloprevotella that are actually having a gradient. And if you come to the lower level of that panel, on the opposite side, you have that predominated by Bacteroides. Bacteroides is again indicative of a Western community. So if you define this figure, it's on the other end, it's the vanished taxa, which basically vanish when you move to an industrialized region. And you have what they call a blossom taxa, or it's a selected bloom of these taxa that are relegated to these societies that are modernized or they're urbanized in that setting. So we are right now in an epoch of microbial depletion, which basically means we are losing out our ecological identity to this industrialization process. And that is happening in real time. It's happening in real time because of modernization, because of the land management practices that includes what you do in agriculture. And these trickle-down effects can be seen across these indigenous communities, which are traditional. And if you look at it spatially also, communities that are lived in Venezuela or in Africa, Burkina Faso or Peru or Malawi and in East Africa, you have this trade-off between what you see in the industrialized community. And that trade-off, again, is represented by the prevalence or the absence of those unique communities that we inherently need to have in us, but we are losing out on that, thereby rendering ourselves potentially susceptible to conditions that are challenged by these vagaries of the environment, which means it could be autoimmune conditions, it could be chronic conditions that are setting in that regard. And how do we try to attribute this exposure of this microbial cloud that shapes our health? It basically trickles down to the way we grow our food. And the way we grow our food in the environment, which actually is going to feed back into how homeostasis is maintained in our gut. So this paper just recently came out in EMBO reports. It's actually a very good illustration on how optimally you grow your produce or your plant. That is actually going to help you recruit the right set of microbes in the way that you consume your diet, thereby training your gut to accommodate for the right set of dietary ingredients that will nurture the microbes that are there inherently. And there are different facets to it, starting with how you practice the agriculture. It could be the conventional agriculture, which is laden with increasing herbicides or fertilizer or pesticides, that is making you fragile. It's making the ecosystem fragile. It's reducing the microbial diversity in these farmlands, which we need to take care of, and thereby also reduce the amount of specific consumption of certain ingredients like processed sugars, increased consumption of red meat or processed meats, and reduce the level of pesticide exposure. Those are concerted things that need to happen, and that need to happen. It would happen only if there is a response from the society for this change, or the request or the motivation for people to look into these elements and unravel further data that can help inform such changes. And from the soil or from the environment, it comes down to this component called the phytobiome, which is actually the most critical set of plant-associated microbes or soil-associated microbes that are helping us feed our own guts. And again, this is constituted of different groups that you find in the environment, driven by the same set of factors that drive microbes in our gut. Starting off with soil pH in this case. pH is a variable that hugely governs or controls the microbes that translocate in us. So same way you have, again, the carbon sequestration capability or redox status, which is actually the oxygen availability or hypoxic prevalence in these soils. That is actually going to trickle down and affect the way microbes are recruited. So if you look at, again, spatial relevance, I'm skipping into the plant system in this case, very specifically looking at spatial partitioning between the environments, the soils, into the plant or the crop that you're trying to grow. Again, you have the spatial divergence, starting off with the root hair, the epidermis, the bacterial translocation or the fungal translocation into regions like the cortex or the inner root. You can find the analogy to the mucus layer that's lining the gut in this case. Again, the same way you have a certain level of translocation that needs to occur for these microbes to be endophytic or internalized into the plant system in such a way that when you're consuming a crop, when you're consuming a vegetable or a fruit, you're going to have the seed of that vegetable or fruit that's going to inherently capture a snapshot of those beneficial microbes that you need to carry forward from generation to generation. So this is also going to happen only when there is a right set of these homeostatic interactions that occur. And I just take one example of an event that happened in China a decade ago. This actually is brought up to me by one of my collaborators, is actually right here. It's an incident that happened in China where they found infants who were fed processed milk that was laced with this protein-rich formula falling sick. They ended up developing kidney stones, and they ended up developing several issues associated with their gut as well as with other comorbid conditions. And 300,000 children were affected. This fact was actually suppressed, and they found later through investigations that these dairy products were tainted with this product called melamine, which is something that is a compound that's introduced into baby formula to show that it is high in protein content. It basically mimics a high protein content drink. It so happened that there was this microbe, essentially a soil microbe, at that time it was called Klebsiella terragina, that had translocated into the gut through the medium of that powder that they added. And that microbe was able to use melamine as a substrate and produce a metabolic byproduct called cyanuric acid. So cyanuric acid became toxic for the kidneys, and this resulted in deaths as well as adverse events for these children. So this is one such odd instance where for the first time they were able to show that in the gut microbiota, there was an environmental microbe that was causing an adverse effect. Obviously, there are separate scattered events associated with, say, salads, or pathogens that are laced in the salads during supply chain processes. But this is a one-off event that is completely attributable to diet that was ingested. And it actually brings us to that question on how do we optimally consume these diets and optimally account for representative groups of these vegetables that you would consume. And something to that extent was actually put out from this project led by my mentor, Rob Knight, called the American Gut. This is actually the world's largest citizen science microbiome project, which is actually ongoing right now. And one of the key elements of this project was to understand how the human microbiome data is actually in convergence with the environmental microbiome data that you could derive. And so one of the key things that they found from this study was the more the number of plants you consume, the more the number of beneficial microbiota that you nurture in your guts is something that they were able to put out. In this figure on the right is a principal component ordination plot that's represented by the distances defined by the unique fraction of the phylogenetic metric. So they pretty much draw an evolutionary tree or a phylogenetic tree connecting 15,000 odd samples that they collected as part of the first cohort. And they were able to draw distances in terms of relevance from the patient samples, or in this case, the volunteer samples, from their associative dietary patterns. It could be patterns associated with whether they're vegan or vegetarian or they're omnivore and things like that, right? So they were able to actually earmark based on these metadata characteristics how that is going to implicate what types of microbes they nurture and how these dietary inputs are able to help them overcome the element of depletion in bacterial diversity. This is something that's ongoing. It's actually expanded to to Asia. And the reason I bring this up is this study wouldn't happen but for the background that went into it. And the background is what I'm going to focus on, which is a project I was involved with from the beginning days called the Earth Microbiome Project, where we systematically wanted to attempt at characterizing microbial taxonomic profiles for different continents or different types of ecosystems around the world. This starts off with soils, glaciers, water ecosystems, and animal hosts. So basically, this is free living and host-associated environments minus the human gut. So that was actually what the overarching goal of the project was. And this resulted in a publication in Nature in 2017, but it was work done over a period of 5 to 6 years that included 500 plus scientists and 27,000 samples were generated. And this is open source work that's widely available for students to use. It's encompassing data from 7 continents and 43 countries. Again, and also data that's accompanied by metadata information or environmental information, which gives you information like species, pH ranges, where it is located, or geopositioning, or where on the planet you can go and look for these types of species. And this actually was the first reference database of bacteria, on this case bacterial archaea, using the 16S approach for our planet. And it's still ongoing, where they have a phase 2 that they're doing functional representation from a select cohort of these samples. So the way we did it was initially doing a lot of crowdsourcing, which means we were working with laboratories around the world to help replicate the same set of standards that were built in the laboratory by the groups that initially were part of this, which is including our group. And a lot of the efforts were specific to developing the protocols, starting off with 16S ribosomal RNA extraction, basically PCR amplification, and sequencing protocols. And these protocols were followed to the dot across all the laboratories. Thereby, it yielded close to these 25,000 samples represented by soils or aerosol or animal gut. And one of the key take-homes from that study was between these samples that we collected from the host of free living environments, we found the soil having the maximum amount of diversity. In this figure, again, on the lower right panel is the unifrac or the phylogenetic diversity information between these 27,000 samples. And if you ask the question as to what is the most diverse group of samples that you collected, it constitutes 2 groups. One is the soil, and also the other group, which is the plant rhizosphere, which is the region closest to the plant's root surface that is actually hyperdiverse or actually in association with the plant host, thereby making that actually one of the most critical zones where we need to protect those environments to nurture those microbes that otherwise are not going to be helpful if you're going to lose that particular habitat. And if you look at the far bottom, is something like animal surface or animal gut, which is totally different from the samples that you collected from soils or from a plant rhizosphere area. So again, we ask the question as to if you represent this by the ribosomal RNA gene sequences, here in this figure on the Y is the number of 16S ribosomal RNA sequences mapped back against host-associated habitat types and free-living types. Again, the things that stand out include plant rhizosphere or soils. And at the same time, there's high variability when you look at habitat like water, which is non-saline. Again, because that is constitutive of fresh water, which can have trickle-down effects from human disturbance or mass gathering activities. So this gave information to the American Gut Project as to how you can answer the question of from the human microbiota to the environment, whether there are feeder effects. Is there a link between the two? So they were able to plot back from EMP data and the AGP or the American Gut Project cohorts, phylogenetic outputs, asking questions as to what are the interspersed groups between these, phylogenetically speaking. And they were able to find certain areas where there was an overlap. And interestingly enough, the further on question they asked in the lower figure here is if you're finding an overlap between the environment and the human gut, how does it perform when you throw in the human microbiome data, which is actually a highly curated version of the data. So they were able to interestingly find in red on that phylogenetic tree as that's addressed B, groups of these phyla constitutive of firmicutes, which is actually something that's predominant in Western communities. I mean, obviously this is a cohort that's only representative of North America. So that's going to be predominant there. And it's also indicative of traits like obesity. So there's further on efforts that are going in this direction to attribute elements associated with environmental feeder effects, and thereby help represent the differences we have as an industrialized community or a westernized community, and how we can go closer to a traditional diet associated community. It's actually a biggest challenge that they're trying to address, and this is something that several of these companies are also trying to solve by generating these traditionally robust probiotics, which is actually another area that's totally a different topic at a different time. But I'll sort of limit it at this point, where we were able to also provide these cartographic inferences between continental areas and how they map back when you look at traits in species and how they can help inform variation. So just diving into the variation aspect. So we are spatially having these interferences that occur starting from the time we are born, from the time we give birth to when a child is born, and then through different developmental stages. And again, it's even in different stages you're going to have variation in this abundance and diversity that's included by foods. And at the same time, as you talk about all these elements, you have a different point in time disturbances inflicted upon you through these healthcare adaptations that are practiced, right? During COVID-19, for instance, one of the biggest paranoia is resulting from the overconsumption of drugs, or in this case, antibiotics, thinking that it's going to help them. And in the US alone, there's close to 2.8 million antibiotic resistance each year, and close to 35,000 of these deaths that happen. And again, there's obvious reasons or several feeder effects to the prevalence of this resistance, starting off with overuse of antibiotics in livestock or farming, and lack of access to hygienic environments, or sometimes in this case, over-hygienic environments, right? In the case of the developing world, it's the other way. And again, poor infection control in hospitals and clinics. It's something that it's very alarming that you see this in the US. So which leads people to ask that question as to why has this country alone had so much of death in terms of comorbidities associated with COVID? So there is potentially a hidden lurking factor here which needs to be accounted for. And I hope authorities wake up to that, and there's some efforts in that regard. So in terms of global mortality, especially due to the AMR or antimicrobial resistance, we are looking at close to 47 million deaths in this case, 4 million in this case, in Asia in mortality, and 300,000 for North America. So this is actually pretty staggering amounts that are distributed across the planet. And the example that I want to take is associated with AMR, because we want to we set up on this effort to study the microbial cloud of the built environment in New York City, starting off with the initial effort that was called PathoMap, which stemmed from New York City, trying to understand spatial resolution of human and bacterial diversity in the city overall. And this is the first city-scale metagenomic profile that was built. It basically adopted a shotgun metagenome profile or approach where we were quantifying abundances from the sequence approach, sequencing done using paired and NGS platforms, and mapping the sequence data back to reference genomes, in this case, pretty straightforward. But what was done primarily was each of these spatial locations across the city of New York were ventured into, and swabs were collected from these surfaces, starting off with the, say, ticket vending kiosks to a turnstile, to the subways or these built environments. In this case, it's a contained environment where people congregate. And in that, what yielded was close to 46.9% of bacteria. And at the same time, a large fraction of that constituted unknown organisms, which is very paradoxical, because we were thinking that the databases are right now pretty robust. Right now, as this is from data from 5 years ago, to actually account for viruses or plasmids, information from plasmids or from eukaryotes that you can represent in a way that is understandable. But we realized that this is unknown, and several of these were things that need to be unraveled. And this is sort of the visual interface that was established, where you were able to ask the question as to what bacteria is prevalent in what part of the city. And this is a map where you give density of Pseudomonas species alone and look at the density in very busy areas in Manhattan, like the Wall Street area or the Midtown area where Penn Station is. So they ask another question as to how does this compare to the environmental data or surface data associated with what you find on the subway surface or these stations compare against. So they compared it to the HMP, and they found this resembles the skin in a very broad scale. And thereby, some of these species that came up are species that you will find in regions like what you find on your skin or your nose, like Propionibacter acnes, or some of these species that can be opportunistic pathogens like Staph aureus. And this led to the evolution, basically the step up of this particular project, where Gates Foundation got interested, and they were interested in funding us with a phase 1 grant for genetics and epigenetics of antimicrobial resistance. So the first approach was to do a concerted metagenomic approach to look for microbial signal, metagenomic signatures from these city-level surfaces. It expanded to close to 60 cities around the world, and the major goals was to provide geospatial resolution using these metagenomic maps that can help inform how people movement or passenger movement is, or exchange of, say, groups from different continents, how they're traveling, or whether they're leaving behind a trail associated to perhaps their skin microbes or the microbes that are specific to antibiotic resistance markers, right? That could also be something that can be derived from that data. And also the other goal was to derive gene clusters that were novel in helping evolve gene cassettes for biosynthetic drug discovery, specific to antibiotics. And this could also become a resource for that. So to that, one of the key studies that came out very recently in June in Nature Medicine was led by Niranjan Nagarajan from A-Star Singapore, and his group actually worked on a healthcare facility, which is actually part of the MetaSub. They call this MetaMed as a subsidiary. And they looked at building the first cartographic map of opportunistic pathogens and antibiotic resistance genes in a hospital environment. For that, what they did was they swabbed the hospital setting over 3 different years, starting in 2017, and they collected like close to more than 400 samples in this case. And this was actually staggering amount of data that they generated from that. It resulted in novel genomes for 2,300 of those samples. And actually they were able to generate phage sequences from 1,600 of samples. And they're also able to show, using this parallel approach of doing direct shotgun genomics and culture-enriched long-read genomics, that there were specific spatial variability between these species that were located in the hospital environments. And starting with the demarcated zones, like isolation rooms for people who have conditions with multi-drug resistance, wards, as well as the standard wards. Isolation rooms are pretty much people who are potentially coming with, say, for instance, a COVID infection. They basically put in that, or a very, very highly virulent infection that they're coming in with. So they were able to demarcate, especially on the abundant spikes that they could find with specific escape pathogens in this case, like Klebsiella pneumoniae or Pseudomonas aeruginosa or Staph aureus, as you know, come under the category of escape pathogens that are highly on the high-risk list that you otherwise don't want in terms of a spike up. This is coming from healthcare facilities, from places where you think that everything is protected, everything is clean, and you're not going to have these nosocomial infections going into these facilities, which actually resulted in the first ever representation of ecological niches within a hospital environment. Here in this case is a spatially mapped out, on the left is basically a Bray-Curtis way of representing species abundance and a species presence or absence overlap between these sites, starting off with the sink trap or the aerator, which is very different from what you would find on the bed railing or the cardiac table or pulse oximeter. It's actually connected to your fingertip. Again, you have species that are highly reported in nosocomial settings. And on B, in the figure B, is the differential abundance of those species, whereby you're able to see species such as Acinetobacter or Corynebacterium, which are completely opportunistic pathogens. I mean, pathogens that are going to cause adverse events. This is something that has not been seen before and was staggering. There's further more detail on that, but I'm jumping straight into the subway where we, every year starting off with 2016, have on one particular day in the year collected samples globally from different sites across the world, starting off with New Zealand in Auckland, going all the way to Alaska. And this is something that has been done consistently, and I launched this in India in 2017 in the city of Hyderabad. And over a period of time, this is actually a readout for samples collected in a 24-hour period from different cities. And right now, we are actually at the culmination of the phase 1 of this project, where in terms of the curated version of what's going into a manuscript that I'm going to talk about, we have 4,700 samples from 60 cities around the world. And we have created this resource called Metasub Map, where you can go onto that portal and actually click on the city that's relevant to the site where it's sampled from and understand what types of microbes are prevalent in that city. At the same time, this also has become a resource for next metagenomic data that is open source and available for researchers to understand if they want to provide spatial representations between a new site that they're sampling from. And potentially this data does not include data from India. So in some of the preliminary results, there's actually there's a preprint out there available online for free, and the manuscript is currently in review. So this actually had an approach for understanding in spatial resolution metagenome data collected from the biggest cities in the world, like starting on New York City, Rio de Janeiro, London, and Tokyo. So we sampled from the subways in all of these cities, one of the oldest subways being in London and New York, and also, and in Rio, they were collected during the time of the Olympics. So you can have the spatial hierarchy or how the representation of microbes are between continents like North America versus East Asian countries. And you can see that spatial variability on the right panel is the Jacquard representation in an ordination plot. And there is a certain transitionary zone in Europe where you have interspersed effects between East Asia and North America that you see. At the same time, you have a separate clustering group that's in between but scattered out, which is from the sub-Saharan African type. And again, interspersed effects are coming in from these continents that are not, that do not commute as much as people that commute from these regions of the world, right? So, and the way this was done also was using a Kamer approach, where there's a breakdown of the raw reads that were generated from these geotag samples, where each of these samples had a GPS information. Each of these samples had some spatial information recorded during the time when it was collected. And some of these metadata tags included where it was collected, like it was a bench, or whether it was a kiosk, or it was a bench, and a timestamp associated with it. So that actually helped render some robustness on how you interpret the data and how relevant it is from the site it's been collected from. This is a visual representation of what the cartographies could look like. It's just a one-shot representation of sites from New York and London and Hong Kong, where you can see this density-based representation of species from these sites. And one of the big takeaways here is from the subways alone, we were able to recover 1,300 novel bacteria just from subways alone. which is actually staggering if you think about this is a built environment. This is an artificial environment where we accommodate ourselves to transit from place to place. And again, this is from metagenome assembled genomes that is found in the metasub data. And again, in this case, the outer black ring or the figure on the left is representative if the MAG is associative of a known species or if it is associated of a phyla of the MAG. So there were a distribution of unknown as well as the known entities there. And that way they were able to actually feed into the database and see whether there are similarities or complete novelness in these sequences, thereby resulting in these number of species. So there's also a phenotypic way of distributing these samples that were found starting with the way they accommodate as a commensal, or they form biofilms, or they are gram-negative in terms of their trait. And this also helped build microbiome diversity profiles, starting off from each continent, and also functional profiles on the pathways that are pretty predominant in these samples that you collected, starting off with respiration or amino acid biosynthesis, or in this case, fatty acid or protein, so vitamin biosynthesis. This is something that we try to understand as well. And if you look at the variance on the top panel of this figure, one of the drastic variants that you would see is between sub-Saharan Africa, for instance, versus what you see in Middle East. Again, so there is a drop, an obvious drop in proteobacterial communities in the samples coming out of Middle East. And there is a predominance of more of firmicutes, which is actually associated with obesity in Middle East samples. Again, at the same time, you have an increase in proteobacterial samples that you find in African samples, versus the same trend line varying between Europe or North America. Again, you can have these spatial inferences that you can make, but there's there's furthermore data that's needed to do a lot of cross-validation between these sites. And the other take-home was the AMR classes. On the bottommost figure, we found that there were AMR classes that were specific to beta-lactams or categories of these antibiotic molecules that were actually having the potential to cause multi-drug resistance. In sub-Saharan Africa was very predominant with these AMR classes. It's actually alarming to see. And also in North America, it was pretty consistently represented across the board, where it was more sparse in Europe or in Oceania or Middle East. Indian samples are really going to be interesting once we overlay this on this, considering we have a uncontrolled way of consumption of antibiotics. And in terms of AMR class, there is also a representation from prevalence of these AMR classes and across the number of sites. On the left figure here is the reads per, on the y-axis is the representation of reads per kilobase per million mapped reads for the samples that have been generated. And this is actually representative of different classes, starting off with beta-lactams, the multi-drug resistance, rifampin, or if you look at these metronidazole associative antibiotics. So each of these have separate classes, and in terms of number of genes represented, the number of genes were most highly represented on the rightmost figure here in Colombia, or at one particular point it's Bogota. Again, if you look at cities like San Francisco, it's pretty much staggered across versus several other cities. This is a snapshot of few cities that I've taken in here, but you can actually have representation of different cities around the world. If you look at something like Santiago, Chile, there's an increase in the AMR genes being locally represented across, and which is actually really alarming. And a city like Ofa, which is second in that category, which is in Nigeria, is also having a consistent representation of AMR genes. Interestingly enough, Ofa was a city in Nigeria where they found a huge proportion of the normal bacteria as well. So it is interesting to note that point as well, because in Africa, there's again uncontrolled use of antibiotics, and I think some of these antibiotic generics have been produced in India and exported to African countries. And in between is this co-occurrence map plotted by Jacquard, where you have the co-occurrence understood by these antibiotic classes across city in terms of the correlation. Again, you have these areas that are specific to particular classes of antibiotics as a visual representation. From the viral DNA sequences that were recovered from these sample sets, what was interesting was we were able to kind of unravel close to 11,000 species specific to viral DNA sequences that was unaccounted for, say, bacterial representation, right? And the way it was done by actually taking into account the CRISPR classes from the subsets of these samples and also mapping it back against the JGI IMG virome database. This is the database I spoke about initially, where they had 750,000 sequences specific to DNA viruses alone. And then after doing a subtraction approach where they were able to filter the species specificity, they were able to narrow down on 10,928 novel viruses just from subways alone in the world. And this sort of is a staggering number that we're seeing across these cities, potentially interlinked by global travel, if you will. And mind you, this is not RNA viruses that I'm talking about, because that is a separate concerted approach that needs to happen. This is just derived from DNA metagenome data. And so this is also something that's right now in the review for that manuscript in prep right now. And I'm skipping very fast into what's being done in India, in Metasub, in Hyderabad. We initiated this in 2016. We collected samples from the Hyderabad metro before people got on board, which means we did it during their trial runs. And the metro was inaugurated at that time. So in Hyderabad, it became the first baseline set of data we collected from Asia. And this was included with some of the sampling efforts that we carried out there. Also got compared sampling from the busy MMTS systems in the city. And these samples are basically still not yet on the Metasub sequencing output, but currently what is ongoing is during the lockdown, with the efforts of my collaborators, Someshwar Sagarthi at Osmania University, we've been able to collect samples during the lockdown, which actually makes it interesting because we are going to have comparative snapshot of samples both before the subway was occupied, during a lockdown, and then after when the lockdown is clear, hopefully, and the people start coming back on these transport models. It's going to have sort of a progression in terms of how these AMR classes would predominate or would succeed based on their specificity. So I think an approach like this can be expanded on a pan-India basis. To that regard, one of the reasons I'm here is to actually start off Metasub in Chennai, where it's not yet being kickstarted, and there are several challenges associated with Chennai right now, notwithstanding the fact that the worst cluster outbreak happened in the city of Chennai, very close to the metro transit center and the bus station from a vegetable market. And that's actually one of the areas we're trying to tackle that from the get-go. So potentially doing this kind of surveillance could help address challenges and address some paradoxes similar to the prevalence of superbug gene that was found originally in New Delhi in the Arctic Circle. I'm taking an example of the New Delhi beta-lactamase, metallo-beta-lactamase incident that originally after 2 years from the time they found that sample in Delhi, they found that in the Arctic Circle in northern Norway, the soils from there were laden with the antibiotic resistance genes associated with that particular plasmid. It sort of gives you a moot point as to ask whether the bug took a ride using a different medium that basically transferred that particular trait into that extreme habitat, right? It so occurs that one of the possible ways this transfer could have happened is because of these migratory birds that go from north to south and south to north. And that's one of those washes that people are trying to address. But perhaps concerted efforts on looking at biosurveillance from such ecosystems or specific pockets where you have human interaction that's going on at a rapid pace could help unravel those patterns. I think that's, again, a moot point that we need to address. Once again, thanks to Metasub team, extensively collaborators Benjamin Young, Christorian, who are doing a phenomenal effort in coordinating several collaborators from around the world, all collaborators in India, Narasimha, without whom Hyderabad wouldn't have happened in Hyderabad, and IIT Madras, ISA Kolkata, Shastra, Pondicherry, the funding agencies here, sponsors, as well as several of these initiatives that have helped and also helped move the research forward. I'd like to acknowledge my mentor, Professor M.S. Swaminathan, who recently celebrated his 95th birthday, and he's actually somebody who I look up to, and I think generations of us look up to. And I just want to quote one of his saying, like quotes from there, agriculture is the backbone of the livelihood security of nearly 700 million people in India, and we need to build a food security in the foundation of homegrown food. And now more than ever, we need to maintain our ecological identity to help cope with this ongoing onslaught of exposure to pathogens and these risks associated with modernization or industrialization. And special thanks again to Nandini and ICER organizers. And I'd like to thank you for your time and attention and take questions. Thank you.
Thank you, Bharath, for that fascinating talk.
Thank you.
Very exciting work and a lot of work, clearly. If anybody has any questions, maybe you can unmute yourselves and ask questions or type it into the chat window.
Yeah, I just want to make 2 comments. I think these were more than 3 seminars packed into one. It was very nice, exciting, because several facets came eventually regarding microbiome in different ecological contexts, correct? Yeah. So now all the countries are chasing development in GDP concentric form, right? They are all GDP centric developmental models.
Sure.
Right. So in the process of this kind of chase, we are to imbalance.
I'm not able to hear. I think it's just 1 second.
Could you repeat that, Professor Rao?
Our ecosystems.
Professor Rao, could you repeat
And
that, please?
This will be very well reflected.
I couldn't.
In the microbiome that you are going to analyze. So at some point there will be.
I missed portions of that question.
Be an issue of where do we balance?
Professor Rao, we can't hear you. Maybe if you can turn off your video, maybe we can hear the audio. Yes.
As well as our growth. Where do we balance our microbiome diversity as well as our developmental models? They are going to be at loggerheads at some point. Am I audible?
Yeah.
Meaningful patterns out of it. Not just quotes now.
Bharath, did you get that?
No, I did not. It was.
We can't hear you now, actually. I can't hear you.
You can't hear me now?
No, can't hear you.
Oh, well, okay.
Your other device has disappeared.
Is it okay now? Just a second. Can you hear me now?
Yeah.
Okay, sorry. Yeah, I think my phone. Yeah, I couldn't hear that question, Professor Rao.
I think he was asking about developmental paradigm and microbiome patterns, and at what point would you use one to inform the other?
Yes, that's a great question. So I think the developmental paradigm is still something that's in synthesis, but there have been flagship studies that have been conducted. I think some of these studies have come from Harvard. Very specifically, there was this cohort in Fiji called Fiji Comp, led by Ilana Brito from Eric Alm's lab, where she showed that this is a group that is an islander group that was completely disconnected from North American cohorts. And they were able to see that some of these individuals who inherently were practicing just their traditional diets, when they got an exposure to import of processed foods, there was basically a very interesting social facet to it, where they talked about the peer pressure of adopting to a modernized realm of dietary consumption, which basically resulted in fluxes in the microbiota or the changes in communities, microbial communities, which is something that's trickling down to different societies. It's actually trickled down in a big way, I would say, even in India, at least among the millennials or the generations younger than the millennials, potentially speaking. So these are the kinds of models that the country needs to be aware of. I know Indian government launched the Indian Gut Initiative with fanfare, but I wonder if there is a concerted way to earmark generating metadata which is specific to each regional attribute, which means the North, again, has very distinct diet compared to the East, the central part of India, or the West, and even the southern part. So there's going to be a lot of heterogeneity in the data that you would generate. And the robustness of that data, or the impact of that data, can only be realized once you have a very plastic model for using the metadata that is provided along with these data sets. Very similar to what the American Gut Project did, although it was not a case control study, it was a citizen science study. But I think we have the opportunity to enforce or even implement some of these practices or best practices that have been adopted in such successful endeavors. And we are really strategically positioned in that way, because as India is so diverse, you have tremendous potential to address it both from the level of the human host and from the level of the environment, very specifically speaking in systems like agriculture. It's actually a very, very strong way. There's an opportunity there to do that.
Yeah, thanks. There is one more question. In Sahara, is there a case of unregulated antibiotic consumption in the Sahara region?
So the Sahara region is why Nigeria comes up. And Nigeria, again, is one of the biggest importer of antibiotics from countries that manufacture generics, which is India included. I think we are one of the biggest exporters of antibiotics. And there is unregulated use of this medication. So much so, because we have one of our collaborators from Nigeria, Yinka, and he says that they don't even have, in some of the areas, a structured way where wastewater is processed, for instance. They don't have regulated sewage treatment systems, which means you're going to have runoff from domestic households, runoff that would include antibiotic traces. It's going to come up from agriculture, it's going to come up from animal feed, from domestic use. Again, it's going to increase that element of spiking, which is going to contribute to the overall increase. But again, as I pointed out, I'm looking forward to having data from India on that map, on that panel.
Yeah.
And that's going to unravel several more things. And I don't know how the regulations are going to control getting that data. Hopefully we can get some data.
Okay.
Thanks.
Do we have any more questions? Would someone like to ask something?
So host genetics also influence the microbiome diversity or population.
Sure.
So my question is this data set, or this robust data set, will be helpful to understand that how host genetic can influence the microbiome data set?
Yeah, that's a good point. Good question. And very specifically, if you're generating metagenome assisted genome assemblies, there could be a concerted way where you are looking at ethnic differences between individuals or communities across these continents, and also perhaps map back into genome data that's been generated from those ethnicities, from systems that have been built up from before. I'm talking about systems like the fancy 23andMe ones, or the ones that are taking into account human ancestry, but at the same time also taking into account some of the metabolic traits associated with them. That could actually provide an opportunity to help map back to host-associated factors. This one such effort that's going on in our collaborator in UK, Eran Elnek, and he's looking into some of these elements in the host factor associative approach. That's where the epigenetics part would come in, which is actually the phase 2 of this project. But I wanted to add to actually the element of environmental microbes, where there's tremendous scope for understanding whether innate immune responses in plant systems. It's possible that you are looking at comparing, say, a wild type plant versus a knockout, was mutant versus a transgenic variety, and ask questions as to whether in this example, like say, for instance, systemic acquired resistance, you have certain immune receptors that you find on the surface of the leaf having a crosstalk with the rhizosphere microbes in the soils. So these are kinds of paradigms that can be built, that is actually available in certain sparse data sets already. And that could really make the whole crosstalk robust, where I talk about this convergence. This is actually the grand challenge. And once these modules are built, the interspersed effect of these modules are built, you can talk about the connection or the convergence between, say, agriculture and human health. Okay, environment and human health is going to be a huge challenge, but I think subsets or modules of that can actually be worked into, and maybe like a regional setting in India also, you can pick up a cohort of individuals from, say, southern India and look at the agricultural or specific land races there that are predominant in that region, handpick them, look at those traits, at the same time also look at the host-associated traits in those individuals and pick up signatures from their gut microbiota. I'm just taking a hypothetical example in this case. But there could be, the scope is enormous. And I said there's a lot of nascent initiatives that are ongoing. And we are literally in the 5th year from when several of these advances in metaproteomics or metabolomics have occurred. So it's a matter of time by which we will have access, at least in India, have access to those technologies. And right now, we're heavily reliant upon these well-funded groups across the globe. Hopefully, there's democratization of that access, and we're able to generate data sets that will really strengthen this field and help people, researchers work together and generate data to that direction.
Bharath, there's another question.
Yeah.
Akanksha again, the one who asked about Sahara. Did you take into account the hygiene conditions of the populations, that I guess the foraging populations?
Oh, yeah, that's a good one. I mean, the foraging populations pretty much. So this is something that, I just want to give clarification on that. This is the work I did not do. This was done by Justin Sonnenberg's group in Stanford. And this is the work done in the Hasda populations in East Africa. So they went into their environments and interacted with the Hasda community people. So they did not interfere with the way they practice their foraging activities or the way they consume their food. So they basically let them be
I think maybe what she was asking is if you took swabs from the hygiene conditions, from how they live, from the environment, apart from just from them, and then kind of.
If I recall, I don't recall that study doing environmental samples from those areas. So those samples are not connected to the meta subsamples. That's also another clarification that I need to provide at this time, because that's a separate study that I basically had to provide as an orientation to the traditional diets. Yeah. But that's an interesting, that's an interesting question for sure.
I think Vijay has
just out of curiosity, this first of all, nice talk and a lot of information there. I was just curious why meta sub, because here you're looking at a moving target, right? In subways or trains, you have population that is moving from point A to point B. So you're not looking at a clean or a local microbiome data, right? You're looking at, and especially if you look at New York or London subways, you have a global population in those subways, really, right? That's one thing. And the second, I was just curious, when you're talking about second generation and third generation people who've moved to developed nations and losing their microbiota, I was wondering, have you considered looking at specific locations like Chinatowns, that maintain a lot of their original.
Right, that's a good one. Yeah, so they in New York City, very specifically, there's been some the ethnicity information that we're trying to track on was picked up from some of the signals that we get from diet-derived data points. And again, you have these locations in Manhattan, for instance, Chinatown, where there are predominant neighborhoods, as well as eateries in that area. So people leave behind traces of food as well that comes along with the data that you can pick up. And one such effort, again, with the American gut, there's a concerted directionality being rendered there, where they're mapping metabolomic potential from the diets that they report from people who say they consume that ingredient, and they were able to attribute that to a specific metabolite associated with the diet they say they consume. If it's brassica or like cruciferous vegetable, they were able to actually map back from the fecal sample to that metabolite. That would give an enriched signal that confirms they did eat that ingredient. And that's also something that could become very interesting in Metasub. But we don't yet have metabolomics in the fray. Once that gets in, it's going to make it really more interesting. So perhaps the phase 2, if funding comes through from Gates, that would really help augment that effort, because phase 1 is just 100,000 and phase 2 is 1 million. So there's ongoing efforts to help convince them that we need to get the funding. But again, there's a parallel effort also to map for COVID, which is also going on the subways around the world right now. So it's this year in New York, it got launched in New York, where we're looking for RNA, collecting RNA samples from the subways to look for environmental prevalence of COVID. Not only in subways, but we're also looking at it in wastewater. We're tracking down on that facet. But again, several elements need to come together. Again, the cost factor is a big challenge that everyone's trying to address. So there are so many possibilities. Again, I mean, the scope is not limiting, and we can go in different directions.
Thank you.
Thanks for that.
Yeah. So if there are no more questions, I'd like to thank you again, Bharath. Thanks a lot for doing this, for staying for so long, and sharing all of this with us. Yeah.
Thank you. Thank you so much. Thank you, everybody. Thank you for your time, and thanks for the invitation again.
You're welcome. Yeah. Thank you so much.
Have a good evening. Thanks.
You too. Yeah. Bye-bye.
Bye.